right, everybody. So in this lecture, we're going to talk about helminths, specifically the roundworms and the flatworms. These are another type of what are often considered as microbial eukaryotes. However, oftentimes these organisms are not do not need a microscope to view them. However, they're classified in microbiology oftentimes because they're spores or cysts or eggs, or however you want to call it, are actually how we identify infection, and those are actually necessary to view under a microscope. And so by the end of this module, you'll be able to identify the major types of helminths, understand how helminths infect their hosts, explain how helminth infections are diagnosed, identify the main cells involved in the immune response to helminth infections, and explain how helminth infections are treated. And so, first off, we'll start with our roundworms. These are going to be like nematodes. They're unsegmented, so they're just one, body segment. They do have a full digestive system, even when they're in their infective stage. In contrast, some organisms do not have a full digestive system, like some of the Helminths. So here, this is just a cartoon showing you that digestive system from the mouth to the pharynx, intestine, all the way to the anus. Probably the most common roundworm infection in the US is the pinworm, which we can see here. This will cause infection in your intestines, and it's got a, you're going to see a recurring trend here is you ingest these eggs in some way, and then they infect your body, and they're going to be in your intestines, and then you shred them, or you shed them through your feces. Oftentimes these organisms are going to be like soil associated helminths, so they're going to come, you're essentially going to get infected by through the soil, right? So you might, like, kids will get this because they'll get soil in their hands, put their hands in their mouths, and then they eat pinworm eggs. Now these can be asymptomatic, or they can cause irritation, oftentimes itching around the anus, because what happens is kind of a weird little thing, is that these female pinworms will travel down to the anus and lay eggs on the anus at night. And so one of the ways that this can be diagnosed, which beyond just microscopy, is that, and you may do this as a nurse, is you take double-sided tape, you basically press that to the anus of the individual that is thought to be infected, and then you give that double-sided tape to the lab, and then the lab will actually, view the pinworm eggs under the microscope. Then we have ascariids. This is another type of soil-transmitted helminth. These can be rather big. This is inside of a Petri plate, about the same size as the plates that we had in our lab. And same basic idea is that you will basically get some sort of soil in your hand. You end up swallowing the embryonated eggs. So these are eggs that have larvae inside of them. And those larvae hatch inside of your intestines, and they will pass in your feces. So oftentimes ascaris can be asymptomatic as well, but it can sometimes migrate into the lungs and cause coughing. And then those larvae are actually coughed up and then re-swallowed back into the intestines, where they can continue to mature. So we've got Toxocara, which are these, the two primary ones are Toxocara canis and Toxocara cati, and so these are going to be roundworms that basically infect your cat and your dog. So these can cause like heartworms. They can be passed from mother to offspring in animals, and they can also, as you can see here, they can infect wild animals and game, or they can infect human hosts, and again through some way of ingesting them. And here in humans, they can infect your lungs, your liver, and even your brain. Then we've got hookworms. So we're just talking about roundworms. Now we're going to talk about hookworms. This is a hookworm that is in the intestines of a dog. And these actually just like penetrate through your skin. So like if you are walking somewhere where there are hookworms, right? So if there have been worms that have been have entered the soil through the feces of an infected individual, and you're walking through, those hookworms can actually penetrate the skin of your foot. And here's an example. You can see this line of a foot that has actually been infected with a hookworm. These are the things of nightmares, to be honest. I don't know why it is that the worms and the other unicellular parasites are so much more like nightmare inducing than the bacteria are to me. So here's another one we've got. This is a whipworm, and you can see it's got this long kind of whip-like tail. And here, same basic story. Eggs are passed in the feces. They're going to develop in the soil, and if they are embryonated, so the worm is basically in the egg and just hasn't hatched yet, and you ingest it, then you end up getting infection. Kind of a little switch on the theme, getting out of like our soil-borne ones. We've got trichinella. This is oftentimes found in undercooked meat, and we can see it. It actually insists into the striated muscle tissue. And so that's where it is in, say, something like undercooked pork. And so if there's trichinella in your undercooked pork or some other undercooked game, then it's going to be in the muscle cells. And then when you ingest it, those worms are going to travel to your muscle cells as well. And here, so the major domestic carrier is is just the regular pig. That's why you'll often hear that you should never eat raw pork. Now, I'm not suggesting that you eat raw pork, but I think that in more recent times, it's less likely that you get infected through pork. A lot of pig husbandry has actually changed over the decades, and housing pigs in cleaner environments is something that's more likely to happen than it was 40 or 50 years ago, although it certainly can still happen. I think where you are more likely to find something like trichinella is through this sylvatic cycle. So if you go eat a polar bear or some other sort of bear, make sure that you have fully cooked meat, right? Not that I expect most of the people in this class to eat polar bears or bear, but like if you do, just be aware that if it's undercooked, you might infect yourself with trichinella. So one of the most common ways to diagnose all of these diseases is by examining stool for larvae and eggs. So when you see these eggs in the stool samples, then you know that that there's some sort of helminth infection or roundworm infection. So this is often done through what's called a fecal flotation. So stool might be placed in some sort of liquid solution, and then the eggs will actually float. So we've got a couple of different types of flatworms, which includes one of everyone's favorites, the tapeworm. These are known as platyhelminths or platyhelminthes. And so there are a couple of different classes. We'll focus in on two of them. The first one are your flukes, the trematodes. And the trematodes have these oral suckers and these ventral suckers. You can't really see it here. It's back this area. These may have a incomplete digestive system. And so they can attach to the lungs. So there's like lung flukes, there's liver flukes, there's blood flukes. And so they can attach to lungs, livers, blood vessels. And they do so with these oral and ventral suckers. And so oftentimes these flukes are going to be transmitted through a lot of times snails. And so the embryonated eggs will be ingested by a snail. And then the life cycle of these worms, they'll basically be released from the snail. In this case, they infect a intermediate fish, and then humans get it through eating undercooked fish. Then there's tapeworms. These are segmented worms, and they have a couple of interesting structures that are worth talking about. They've got this scolex head, right? So you've got these little spikes. These things will help attach to the intestinal walls. And I went a little bit out of order, sorry. So then we've got these proglotid segments. These can be released in the feces. And we can identify tapeworm and fluke infections oftentimes, again, through stool samples and through viewing these cysts or these eggs. And for tapeworms, you may also be able to identify these segments. So tapeworms oftentimes, again, come from eating undercooked meat. So if you ingest one of these organisms, or if you ingest a steak or a pork chop or something like that, and it has the like the developing tapeworm in it, then you can actually develop a tapeworm infection yourself. So now, not all of these are soil transmitted, but symptoms of soil and meat transmitted helminths, I should say. Many of them are actually asymptomatic. You may not notice that you have any problems. When they do become symptomatic, they often result in things like abdominal pain, diarrhea. You can have severe things like rectal prolapse. In kids, you can have physical and cognitive growth delays. For some of them, you might have like anemia, you might have like low or loss of blood, loss of protein, you might get fatigued because of that. And so all of these are possibilities. For something like a tapeworm, you might actually have like really low nutrition because the tapeworm is basically stealing all your nutrition from you. Here we go. That's another one. You got symptoms of tapeworm. You get things like upset stomach, stomach ache, a lot of gastrointestinal type issues. You might have weight loss, diarrhea, gas, very hungry, very in need of salty foods. For Trichinella, this one's the one from like undercooked pork. Fatigue is really common, abdominal discomfort, fever, nausea, diarrhea, some of the classics. Later on, you might get things like chills. You might develop a cough. You can get swelling in the face and eyes, aching joints and muscle pains, itchy skin. Now, kind of a not that I expect you to memorize all those things. I'm just kind of like giving you a basic overview of some of the symptoms you might see with these different types of infections. One of the major immunoglobulins that respond to helminth infections is IgE. This region up here, this FC region, is what binds to your helminth, and this kind of marks it for attack. And so here's a video of a bunch of eosinophils attacking a helminth. Looks like a roundworm. And so what you can see in this process is you get the swarming of eosinophils. What you can't really see is those eosinophils degranulate as well. So they're basically like releasing these granules that poke holes in the cell membrane of this worm. And so you can see that there's an initial like attack, and then there's just this huge migration of eosinophils towards this this worm, and they are just tearing it to pieces. It's pretty interesting. And this is what happens if you have an infection with a helminth. All right, let's go to the next slide. So there are a couple of different types of drugs that might affect, that you might be given if you have a helminth infection. You might get something like praziquantel, which changes membrane permeability, or ivermectin, which kind of came into popular culture through potential COVID treatments. Then you've got things like albendazole, which prevents the polymerization of tubulin, and so does mebendazole, which does basically the same thing. And so those are like kind of interesting things, because keep in mind that our cell membranes are similar to the cell membranes of these other eukaryotes. We also have tubulin in our cells. So taking that in mind, what special considerations need to be taken when treating eukaryotic infections in relationship to the infected organism? And so another way to think about that is, when we're making a drug to kill, say, a roundworm, what do we have to consider about that roundworm and about us, the thing that's infected with that roundworm? Take a moment to think about that. Pause the video and start back. Sorry about that. Welcome back. I don't know why I actually paused it on my end. I don't know what you saw when that went out. But so what I wanted you to think of here was the idea that since we are also eukaryotes, that we're, whenever we're taking a drug that kills eukaryotes, it's very important that we make sure that that drug is not toxic to us, because we have, tubulin, we have similar cell membranes and those sorts of things. And so hopefully that's where you were going with your ideas. With that, that's the end of the lesson, and I'll see you in class.